Welcome to ACR On Air, the official podcast of the American College of Rheumatology, where we have engaging conversations with leading rheumatology professionals on topics such as the latest research on rheumatic diseases, solutions for addressing practice management issues, legislative policies impacting patient care, and more. Here's your host.
Welcome back to another episode of ACR On Air, the official podcast of the American College of Rheumatology. I am your host, Dr. John Hausman. Show notes for this episode can be found at acronair.org. You can find our previous episodes on Apple Podcast, Spotify, Google Podcast, and on your favorite podcast app. It's not every day that I get to interview a living legend, but today really was my lucky day. You're about to hear my discussions with Dr. Dan Kastner, who's been called the Father of Autoinflammatory Diseases. His discoveries have taught us a great deal about how the immune system works, both in health and disease. And his work has contributed to finding effective treatments for conditions that previously caused significant suffering and early death. While we discuss many of the autoinflammatory diseases for which he's famous, including familial Mediterranean fever and vexus, we go behind the scenes about how he's been such a successful investigator throughout the years. Dr. Kastner talks about a realization he had in medical school that has shaped his entire career. He discusses how he formed collaborations from around the world, challenges that he faced, skills that he obtained, and surprises that he encountered along the way. As always, he does this by retelling incredible stories with great humor and humility. Dr. Dan Kastner obtained his A.B. summa cum laude in philosophy, of all fields, from Princeton University in 1973, and an M.D. Ph.D. from Baylor College of Medicine by 1982. After completing internal medicine residency and chief residency at Baylor, Dan moved to the National Institutes of Health in 1985. He is currently an NIH Distinguished Investigator, Chief of the Inflammatory Disease Section, and Scientific Director Emeritus of the Division of Intramural Research of the National Human Genome Research Institute. Dan's group proposed the now widely accepted overarching concept of autoinflammatory disease to denote disorders of the evolutionary ancient innate branch of the human immune system. Dan has won a number of national and international awards and honors, and really, I wouldn't be surprised if he receives the Nobel Prize in Medicine one of these days. Enjoy our discussions. Dan, welcome to the show.
John, thank you so much for having me. It's a real honor and pleasure to be here today.
We've been wanting to have you on our show for many months, and I'm glad we were able to make this happen today. We have a lot of questions that hopefully will get our listeners to learn a little bit more about you, more about autoinflammatory diseases, and more about careers in research. And I hope that this episode will be useful for a lot of people. I just wanted to get started a little bit about your background, certainly asking about early experiences or influences that made you want to pursue a career in medicine?
Well, that's a great question. And I must confess that, in fact, medicine was not something that I was dreaming of doing through my childhood and adolescence. And in fact, I grew up in a small town nearby Buffalo, New York, in a working class family. And really, my attraction to medicine was more just as one of the possible careers that I could pursue to have a respectable life after graduating from high school. That was it. It was not as if I knew someone who was a doctor or had some early life experience that attracted me to medicine. In fact, when I went to college, I majored in philosophy, which some people would consider to be a totally unlikely major. But I will tell you that actually majoring in a subject like philosophy is great for a career in research. And the reason for that is because, number one, it makes you think about all sorts of broader conceptual issues, which can come to bear, with regard to a career in research, thinking about scientific disciplines. So that's one thing. Another thing is that it does give you a lot of experience writing. And certainly writing is a major part of a research career, no question about it. One other thing that I will mention in my childhood, which may sound preposterous, but is true, fact stranger than fiction, and that is that in 1963, when I was a mere 12 years old, our family was subscribing to that important medical journal, Life magazine. And on the cover of Life magazine was this depiction of a molecule of DNA. This was right at the time when Marshall Nierenberg had figured out the genetic code. It wasn't when the structure of DNA had been identified, which was back in the early 1950s, but the genetic code was considered to be a major accomplishment. And back in those days, it would actually make it to the cover of a magazine that made it to lots and lots of households across the country. And so I was mystified by that, not to the point where I wanted to go out and become a molecular biologist, but certainly that was in the back of my mind, that this was a really neat and interesting thing to do. So you have the event of Life magazine back when I was 12 years old, going to college and majoring in philosophy, wanting to have some respectable profession, and then landed up in medical school. And actually, because this was a relatively non-specific kind of preparation, I was sort of a tabula rasa at the beginning of medical school. That is to say, I really didn't know what kind of a doctor I wanted to be. In fact, the first year of medical school at Baylor Medical School, which at that time was all of basic sciences crammed into a single year,
Yep.
it was awful. In fact, I really didn't like my first year of medical school at all, to the point where actually I decided I was not formally in an MD PhD program when I started medical school, but I decided that I was so interested in immunology and it seemed so fascinating to me that I would take some time off to work in a lab to learn a little bit more about it. And that's what actually got me involved in a research career. Then, at the end of that, when I, in fact, completed the requirements for a PhD, I went back to the clinical rotations on medical school. And that was when I really fell in love with patient care. And that really became a important defining time in my life where I really recognized the importance of putting together the research side of my life with the clinical side of my life. And I really made a vow to my then future wife, Margaret, that that's really what I wanted to do in life, was to find some sort of existence where I could tie together the clinic with the laboratory.
It's amazing. And 50 years later or so, I'm not going to count exactly, but that's certainly remarkable how you've been able to do that throughout your career, is sort of putting the clinical aspect in your patients with autoinflammatory diseases with all the research that you've done. It's amazing that you had that idea, way back in medical school. So you finish your medical school, you do residency at Baylor, and I believe you're a chief resident, and then you're looking out to see what you're going to do with your life and your career. How did you end up at the NIH?
Well, that's a great question. Of course, this was back in the early 1980s that we're thinking about right now. Back in the early 1980s, things were a little bit different from the way they are nowadays. That's 40 years ago, roughly. And so back at that time, the academic medical research enterprise was not as fully developed then as it is now. And so things were a little bit more NIH-centric in terms of building a career in research. There were a number of the faculty members that I knew Baylor Medical School who had done their training at the NIH in microbiology and immunology, at least part of their training at the NIH. And so certainly I was encouraged by them to think about the possibility of the NIH. The NIH also offered the opportunity, which I just alluded to a moment ago, to really combine the clinical with the basic, the fellowship in rheumatology, which is what I decided to do a fellowship in was one where one actually spent a significant part of the time in the laboratory. And in this particular case, the laboratory would be studying diseases like lupus. So there was sort of a natural progression there. Nowadays, of course, maybe in part because of the success of the intramural program of the NIH, there are great research programs that allow the combination of clinical with laboratory research all over the country. But certainly back at that time, the NIH was one of the major magnets for doing that kind of thing. And since that was my already espoused objective, the NIH seemed like a great place to go to do that.
And then as you started working at the NIH, how did you stumble upon working on familial Mediterranean fever? This very little known condition, probably at the time, there weren't that many patients diagnosed with FMF. How did that happen?
It was really sort of like snatching victory from the jaws of defeat. So I came to the NIH and I started at least the laboratory side of my rheumatology fellowship working on lupus. And actually, the project that I was working on had to do with immunoglobulin gene usage, first of all in patients with lupus, but in a mouse model of lupus as well. And so the idea at that time was that there was some sort of skewing of the immunoglobulin repertoire, both in patients and in mouse models of lupus. And so I developed this fairly elaborate mouse system for testing that hypothesis that there was skewing of the repertoire. It took 2 years to develop this very complicated system. And what I found was that in individual lupus mice, there was, in fact, skewing of the repertoire, but there was not systematic skewing of the repertoire across all of the mice in any particular lupus strain. And so I had asked the question. I had come upon what seemed to be a definitive answer. And rather than having the answer, yes, it's true, and now I can spend the rest of my life figuring out how and why and when it happens. Instead, the answer was no, that's not actually the way things work. And you can write a paper about that, but that's it. It's all done. So in any case, in the background, what had happened was early during the clinical part of my rheumatology fellowship, in our new patient clinic at the NIH, we saw referrals of patients with various unknown conditions. And it just happened that I saw a young man in his early 20s of Armenian ancestry who had for his whole life episodes, roughly monthly episodes of fever with mono or quasi-articular arthritis. I didn't know what this guy had when I first saw him, but it just so happened that there was another fellow in the lupus lab that I was working in who was from Israel. And when I told him the story of this very interesting patient that I had seen in clinic of Armenian ancestry, he said, "Oh, Dan, we see this all the time in Israel. That's FMF. That's familial Mediterranean fever." And he was right. We witnessed an attack, aspirated a joint. The patient had lots and lots of polys in the synovial fluid. And then we started a therapeutic trial of colchicine for him. And he responded beautifully. And it's like Androcles and the lion. This guy is still, a fast friend years and years later because I had come up with something that prevented him from having episodes. So in any case, so that was great. But then you put that together, that exciting event of making that diagnosis and making a patient better, with the fact that 2 years into my fellowship, all of a sudden, I was at a brick wall, a dead end with regard to my lupus project. And so I was thinking, well, now what am I going to do 2 years into my fellowship? Well, the lupus project had at least gotten me interested in the genetics of rheumatic diseases because, of course, we were studying immunoglobulin genes and their usage in lupus. And so I got to thinking, well, is there some other genetic thing that I could do? And this happened to be right at the time that the Genome Project was just getting underway. And so it actually was conceivable that one could go after the gene that is mutated in a human disease more easily than one could do that in mice, because the Genome Project was being directed towards developing maps in humans, not in mice, at least early on. So I thought, well, if those other guys, Francis Collins and Lapchi Choi and so forth, could be going after the gene for cystic fibrosis, which is a recessively inherited disease. And they hadn't found it yet, but there was a lot of excitement around that. Then, by golly, Dan Kastner can go after the gene for familial Mediterranean fever, too. I started out actually in a very, very ridiculous way. Since the patient was of Armenian ancestry, I went to the local Armenian church and I met with the priest of that church, told him of my interest in finding the gene that causes familial Mediterranean fever. He was a little puzzled by all this, but he agreed that we would compose a letter that I would write in English, he would translate into Armenian, and then we would put it on one piece of paper, front English, back Armenian, and then I would send it out to all of the members of the parish. He gave me stickers, address labels for all of the members of the church. And my wife and I, on a weekend retreat to the Shenandoahs, stuffed 1,500
Wow.
envelopes to
Oh my
send
goodness.
these letters out to the parishioners of the local Armenian church. Well, I got lots of responses from various parishioners that they were Armenian and had hypertension. They were Armenian and had diabetes. They were Armenian and had all sorts of other things. But unfortunately, not a single case of familial Mediterranean fever.
Why at the time did you already suspect the genetic component of the condition?
Well, it was actually known from segregation analyses that the disease is recessively disease. Nobody knew what the gene was or even what chromosome it was on or whatever, but it was at least known that it was genetic. And that was what sort of got me enchanted with this idea that if they're doing cystic fibrosis, I can do familial Mediterranean fever.
Got it.
But then what happened when I was actually at an Armenian church bazaar one weekend with my Israeli friend and trying to, again, recruit families with FMF unsuccessfully. And he said, well, I can put you in touch with somebody that runs an FMF clinic in Israel. And he has a clinic with like 3,000 patients with FMF, and he might be interested in collaborating with you. So back in those days, we had typewriters. I typed up a letter. I put it in an envelope. I sent it off to Israel, not knowing for sure if I would ever hear an answer. And then a month later, it was incredible. The letter came back. I opened the letter. It said, "This is the letter that I have been waiting for my whole life." This is Mordecai Pross, the guy that I had written to, had also thought about the idea of going after the gene for FMF. He had tried to persuade some of the people working on cystic fibrosis to change and work on FMF. They had not. And so he was delighted that someone was actually approaching him with the possibility of working on this. So he said he would collaborate with me under two conditions. The first condition was that I would come to Israel myself and draw blood on some of the patients and their families. And the reason that he requested that was that he had actually gone out drawing blood on families and sent them to these CF, these cystic fibrosis labs, and they had just put the blood in the freezer and never did anything with it. So he wanted somebody to have a stake in it. And the other thing was that he wanted one of his residents in internal medicine to come and help me at the NIH, which I was happy to have happen. So in the summer of 1989, I spent a very, very memorable time with Mordecai Pross, seeing patients in his clinic and driving all over Israel, visiting people in their homes. Oftentimes, they were thrilled that someone was interested in their condition. So we'd have lunch with them. We'd get the consents signed. Everyone would roll up their sleeves and we would get blood samples on people. So it was fantastic, a totally memorable experience and a great way to get the project started.
And then you came back to the NIH with, I can't imagine a whole cooler full of blood samples. And how did you start trying to figure out where the gene was and try to decode the gene and then figure out what it was doing?
That was an adventure in and of itself, too. So actually, what I came back with was not a cooler of blood samples. Back in those days, in order to get DNA from individuals, one made EBV cell lines. And so during the time that I was in Israel, I had made 347 EBV lines. For each person, I had 3 flasks, T25 flasks, which are the smallish tissue culture flasks. And so I left 1 flask for each person back in Israel. This is sort of like a Noah's Ark kind of thing. And then the other 2 flasks I put into cardboard boxes. And 347 times 2, that's like nearly 700 flasks that I had in cardboard boxes. And I didn't want to lose them or have them in any way damaged or something like that. And so I actually carried them with me into Ben Gurion Airport. And of course, back at that time, this aroused great suspicion. There were machine gun toting 20-year-olds checking to make sure that no one was doing anything suspicious. Well, it's pretty suspicious when you're carrying, these boxes of flasks with you. Fortunately, Mordecai Pross came with them and explained that it was okay. And that wasn't, any sort of malprient or anything like that. So anyway, I carried them back with me. And then what one had to do was actually to grow these EBV lines up to much larger volumes. I had roller bottles of EBV lines from people to extract the DNA. And then the gene mapping was actually done with markers that required southern blots. And to do the southern blots, we had these gigantic sponges that were like 3 feet long and 6 inches deep. We had these sponges in these Tupperware containers so that the transfer fluid would come up through the sponge and allow for the transfer of the DNA from the gel to these membranes. And then we had markers that we used that I had to go to Utah to get from Ray White, who was the person that had developed a whole bunch of markers at that time. But there were only, at that time, if you can believe this, about 300 markers where their chromosomal location was known throughout the human genome.
Wow.
Throughout the human genome. So it could turn out that the gene would be in some place in the genome where there was no marker near enough to it for you to be able to detect linkage. So it was a real gamble doing this whole thing. And it actually took us from the summer of 1989 to late in 1991 for us to find the chromosomal location as chromosome 16. And then we published that in the New England Journal in the summer of 1992. That only gave us a rough approximation of where the gene was. But then was really the next adventure. And the next adventure, of course, was, well, once you know what chromosome is the gene is on, how in the world are you going to figure out what the gene actually is? So that required actually forming alliances with people that were very, very deep into the Genome Project. And it turns out that in those days, the Human Genome Project was divided between the NIH and the Department of Energy. And it was actually divided according to chromosome. And it just happened that chromosome 16, the chromosome where the FMF gene is located, was actually a Department of Energy chromosome. And so I made a number of collaborations with the major centers for chromosome 16. One of them was actually Los Alamos National Labs, where the atomic bomb was developed, although it was a separate part of Los Alamos for that. And then there was a big group in Adelaide, Australia. And so I ended up traveling to Australia, traveling to Los Alamos, and there were these chromosome-specific libraries of chunks of DNA so that one could make maps to narrow the region down. So there was a lot of work that went into just the physical mapping of that area of chromosome 16. We became sort of like the Genome Project for that part of chromosome 16. We were able to narrow things down to about 200,000 base pairs, and then we had to discover all of the genes that are encoded in that 200,000 base pairs in order to figure out which one was the FMF gene. Turned out there were 10 such genes in that interval. And in screening them, it turned out to be the 10th of them that actually had disease-associated mutations in it. By then, it was actually a race to the death, because what had happened was that there was a French group that had decided that they wanted to go after the FMF gene as well. And they had panels of families that they had gotten from various clinics. And so we and they were racing to see who could find the gene first. And back in those days, when you're looking for a disease gene, whoever finds it first gets all of the credit. And then the other group, if they report it even a couple months later, one can always say, well, they just heard about it from the first group, and so they don't get any credit at all. So it really was important to be first, or at least tied for first with the other group. So I can remember back in June of 1997, when we were on the verge of finding it, that we had heard from our collaborators in Israel that someone's hairdresser had told them that the French were onto it. And at that point, we didn't know for sure what the gene was. I can remember having an emergency meeting of my lab on a Saturday in order to sort through all of this. And actually, the gene that it turned out to be, we had looked at and had ruled it out because of technical issues that I won't get into. But in any case, we re-looked at things, and fortunately, we were able then to determine that that was the causative gene. We ended up publishing our gene in Cell in August of 1997. The French had submitted their paper to Nature Genetics, which only publishes once a month. And so they actually published in the September. It was the September issue of Nature Genetics. But for all intents and purposes, it was a tie. And fortunately, both groups found the same gene as being the causative gene. So all was good in the end. Everybody got credit. Nobody ended up having their career ruined. But it was a very, very exciting adventure, to say the least.
If I remember correctly, the French named the gene, what is it, meronostrin?
Correct, yes.
And you guys named it pyrin.
And so the reason for that, we had a big discussion about that, John. We knew that the French were going to come up with some name. We didn't know what it was going to be, but we were trying to be strategic about all this. Because you see, if there are two competing names, it's just going to be the name that is the easiest to remember or to spell or to pronounce or whatever. So we were looking for names that were relatively short, easy to pronounce, and that maybe you could remember because it had something to do with the disease. So we were thinking pyrexia. At first we were thinking pyrexin, but then we thought, well, that's too long. Maybe, that would be hard
to spell. Cut it down even further.
Yeah, so we trimmed it down to pyrin. Meronostrin, that's a great name because it's named after the Latin for the Mediterranean Sea. But on the other hand, pronouncing it, spelling it, how many people take Latin nowadays? I mean,
Very smart. I like that marketing aspect of your research, that you were already thinking about what name would stick. And it sounds like that was just the beginning, right? Because after discovering the pyrin, the gene that causes FMF, it sounds like very quickly you sort of got hooked on this aspect that you decided to call autoinflammatory diseases. And over the subsequent years, discovered many other conditions that sort of behaved in a similar way as FMF, such as, TRAPS and CAPS and all these acronyms. Can you tell me a little bit about how you started to understand the immune system and then looking for these diseases and then, cloning the gene and whatnot? How did you make that happen?
Well, John, it's really going back to basics. And the basic premise, the basic principle here is what I had explained to you a few minutes ago back in medical school. And that is that I really wanted to have a career in which I was bridging the clinic with the laboratory. And so at the time that we had found the gene for FMF, it was like it was a crossroads in my life. And I could easily have decided that I was just going to confine myself to the laboratory like a monk and be studying the FMF gene and the protein and make mouse models and so on and so forth, and restrict it to a very laboratory-based analysis. And that would have been totally fine. And many people at that time who were discovering disease genes were doing something like that. But really, I felt that it was very important that I liked seeing patients, that it was almost like my obligation to see patients, because after all, I had spent from 1989 to 1997, that's 8 years, looking for it at the public expense, at the taxpayer's expense. And so certainly I ought to at least be willing to see patients with FMF at the NIH. And so I said, okay, I want to do that. But then what happened was that I started getting referred patients, some of whom fairly clearly did have FMF by history and other things that one can look at, their responsiveness to colchicine, et cetera. But then there were some of them that didn't seem to have FMF. They had recurrent fevers of various sorts. There was a subset of them that seemed to have a dominantly inherited condition rather than recessive, as is the case for FMF. They had prolonged attacks of fever. FMF attacks usually last on the order of 1 to 3 days. Some of these patients were having attacks that would last a month or 6 weeks or whatever. They had some somewhat different clinical manifestations, although similar to FMF, but they didn't respond to colchicine, whereas they did respond to glucocorticoids, and their ethnicity could be from anywhere. So we thought that there was something different, and ultimately, one of my former fellows, Mike McDermott, mapped the gene to chromosome 12, came back to our lab to see if maybe we could do the same thing we had done with FMF with this condition and narrow the region down and find the gene. So he and Ivona Aksentievich in my lab had this candidate region on chromosome 12. And we decided that we would apply the embarrassment principle to the candidate region. You see, there were, by then, in 1998, there was a pretty good map of genes of the human genome. And so we knew some of the genes anyway that were encoded in this region of chromosome 12. So we just said, well, what gene would it be that it would be the most embarrassing if we spent 8 years looking for that, as we had to for FMF? Which one would it be that we'd really be embarrassed that people would say, well, why didn't you just look at that in the first place? So the gene that came to our attention in this interval was the gene that encodes the receptor for tumor necrosis factor. And we knew that tumor necrosis factor is a mediator of fever and inflammation. And so, you can imagine that maybe a mutation in that receptor gene could somehow give you a periodic fever syndrome. So that was the first gene that they looked at. And I can remember I was actually on the ward service that fall. And on Thanksgiving Day, I came in to round on patients at the NIH. And Ivona and Mike, the two of them, had come in to check their sequencing gel. And they had actually found mutations in several families. They were the first mutations that we had found, several families that had dominantly inherited fevers. And since it was the TNF receptor, we decided to call the disease TRAPS, the TNF receptor-associated periodic syndrome. So here, within the course of about 2 years, we had found a couple of genes that are mutated in diseases where patients have recurrent fevers, but they don't have any autoantibodies, or at least not very high titer autoantibodies. At this point, this is sort of a whimsical kind of thing. We were on the 9th floor of Building 10 of the NIH. Down the hall from us were other people that were working on lupus and myositis and other autoimmune diseases. And they would always ask me, well, Dan, why is it that you're working on these rare, obscure, periodic fevers and you're not spending any time on autoimmune diseases, which are really the important thing? And so I thought, well, we'll fix them. We will propose a new type of disease that we are studying that is in contrast with the autoimmune diseases. These are the autoinflammatory diseases that don't have autoantibodies or antigen-specific T cells. So that
again, amazing branding, Dan. From Pyrin, autoinflammatory diseases. It's great. Yeah.
So in any case, it was kind of a whimsical thing. And, we submitted this paper to Cell on finding this gene, and they liked this idea and actually put it on the cover of that issue of Cell. But at first we thought it was just kind of, well, that was fun, but whether that will actually turn out, who knows? But then, we got thinking about it that, well, maybe there are other diseases that are autoinflammatory as well. And so we wrote this paper for Current Opinion in Immunology in which we speculated on all of the different known diseases that maybe are autoinflammatory. We didn't have any idea what the molecular mechanism of them would be. We just speculated that these are diseases where patients have seemingly unprovoked inflammation without autoantibodies or antigen-specific T cells, and that maybe they're genetic. In some cases, it seemed like they were from segregation analyses. In other cases, they would be polygenic. And maybe there was some sort of functional connection, but we certainly didn't know what that was at the time that we wrote that paper. These stories that I'm telling you, I mean, it's just like living in Wonderland or whatever. So then a year later, Hal Hoffman at University of California, San Diego, comes out with this paper on Muckle-Wells syndrome and familial cold urticaria. And those were two diseases that we had listed in our article on autoinflammatory diseases as genetic conditions that might likely be autoinflammatory and somehow connected with some of the other diseases on the list. And it turns out that he found the gene that's mutated in those two diseases, in part by virtue of the fact that the gene has a pyrin domain. It has a domain in common with the FMF gene. And so then all of a sudden you have this molecular connection. And what's more, it was just beginning to be recognized by the late Jörg Chopp in Sweden that that gene that was mutated in those two diseases regulates IL-1. And so that then gave a possible molecular mechanism. And there was even one IL-1 inhibitor on the market that one could test in patients that have diseases where there's mutations in this gene. And so all of a sudden you have this virtuous feedback where you discover a gene, you figure out a mechanism by virtue of that gene, and it predicts a treatment, which then turns out to work in the patients. And what was, I guess, what allowed us to then have fairly rapid expansion of the firmament of autoinflammatory genes and diseases was just that, number one, we were early on. It was early days in terms of figuring out genes that cause diseases. And so, we were at a good time to be doing that kind of thing. Secondly, the basic science certainly was going great guns in parallel with what we were doing. And so we could draw upon that. We could draw upon what the pharmaceutical industry was doing, the biotech industry was doing as well. And we had some kind of a concept that we had put forward that turned out to tie all of these things together. Of course, at least at the beginning, it was a whimsical concept, but it turned out that there was something to it. And so it was fun. It was like being kids going into the candy store. And deciding on, whether we wanted M&Ms or Reese's peanut butter cups or what have you. So it
amazing. Over the next few years, it's like hit after hit, home run after home run of you and your team discovering all of these diseases. And it sounds like everything was a success. And I just wonder, in remembering those days, were there any challenges or obstacles that you may have faced? And if you could discuss how you overcame that.
Yeah, well, that's a great question. And first of all, let me just say that, although it seemed perhaps to many people like everything was a success, and I can remember giving talks where at the end of the talk, someone would come up to me afterwards and say, well, for the 5% or whatever of cases where you don't find a cause, I have this suggestion, and then I would have to, say, well, actually, we don't have a 95% success rate. It's more like, a less than 5% success rate. So in fact, there were many, many cases where we did not find the causative gene and still, for a lot of them, have not found the causative gene. So it sounds great, but of course, it's kind of like gambling, or something. And as long as you hit the jackpot every once in a while, there is some element of, I hope you don't consider me immodest in saying this, but there is some aspect of knowing how to take something like that and turn it into a good publication, something that people will understand and recognize and so forth. So there is a trick to that too. There's no question about that. You can have a relatively low success rate and still appear to be successful, even though there's a lot of unsolved mysteries there. I would say that certainly just in talking about that phase of things, there certainly were just many, many cases where we did not have the answer. And in some cases, anyway, it was just a matter of sticking with it. Technology advances over time. A lot of the work in human genetics is very, very dependent upon what technologies you have available to you. I described the early days when we were making Epstein-Barr virus transform cell lines and extracting DNA from roller bottles of cells for each person and southern blots on sponges and so on and so forth. And I'm sure that young people listening to that, would just say, my goodness, that's, they really did that. And nowadays, of course, it's much easier and faster in some ways anyway, and you can be much more efficient with things. And so patience is also an important aspect of this. Patience and optimism.
Mm-hmm. I think you seem to have plenty of both. Looking back on sort of autoinflammatory diseases in general, some people might argue that, well, in my clinic, that just counts for a handful of patients. How has your work helped us to understand anything important in medicine in general, right? And the question is, does the understanding of autoinflammatory diseases help us to understand how the human body works in a better way so that we can also improve the care of people with other conditions?
Well, I would say emphatically, yes, it does inform our understanding of human biology. No question about that. And so just to go back to the early days when we and other people were finding mutations in what we call pyrin and cryopyrin, which is the gene that's mutated in Muckle wells and cold urticaria. These genes turn out to encode proteins that are the building blocks of the innate immune system. And this is like the so wonderful thing that here there is this connection between patients that have various inherited disorders and the building blocks of an important part of the immune system, and that one could actually discover those building blocks by seeing patients, recognizing that they have genetic diseases, and finding the gene. That's a dream come true for me in terms of being able to tie together the clinic with the laboratory. So certainly in that regard, the autoinflammatory diseases have been very, very informative. Another way that they have been informative is that a lot of the genes and pathways that we're talking about are involved in other more complex diseases than these monogenic diseases that are relatively rare. And someone can say, well, gee, when's the last time I saw a patient with familial Mediterranean fever or TRAPS or what have you? And that's a legitimate complaint, or whatever. But on the other hand, we know, for example, that a disease like gout, which is very common, also involves some of the same pathways that are involved in these IL-1 mediated diseases. And in fact, I will say, again, not wanting to break my arm by patting myself on the back, but, in that original paper where we were speculating on what could be autoinflammatory diseases, we did suggest the crystalline arthropathies, not knowing that it was the IL-1 connection or whatever that might in some way be connected with that. And so it's great to see that has led to the use of IL-1 inhibition in some patients with gout. And of course, you're probably aware of the famous CANTOS trial that was done a few years ago, in which canakinumab, another inhibitor of IL-1, was tested in patients who had had a myocardial infarction to see if it would prevent them from having another myocardial infarction. And in fact, there was at least a statistically significant modest reduction in second heart attacks in individuals who were treated with canakinumab. And as a secondary finding, that study actually determined that there was a reduction in lung cancer mortality as well. So definitely IL-1 plays an important role in other things, and a number of the other genes and proteins that are involved in autoinflammatory diseases have a broader scope as well. So it's a way of teaching us. Sir William Harvey said something, I can't quote exactly the 17th century English the way he said it, but something along the lines that nature teaches us about biology the best through the rare exceptional cases. I think there's some truth in that.
Interesting. And in the past 20 years, since you've done work with FMF and over 20 years in Pyrin, has anything surprised you?
Well, I think that one of the things that has really surprised me was a relatively recent finding from our lab that led to the discovery of a disease that we call Vexus. It's another example of what makes the world go round. And so what makes the world go round is young people who have new ideas that are totally at odds with their mentor. So in this particular case, enter to my lab David Beck, who was a human genetics fellow at the NIH at that time, and he wanted to study the genetics of inflammation. But he felt that our success rate was not up to his expectations. He wanted to try a different way of going after human disease genes. You see, the way that we and most people would approach it would be the so-called phenotype first approach, where you would collect together a bunch of patients, related or not related, some of them in families perhaps, that have a particular phenotype, a particular kind of arthritis, a particular kind of inflammation, what have you, but a phenotype that would tie them all together, and then try to find a gene that's mutated in common amongst those people. Phenotype first. Traditional classic way of doing things, and the way that we had been doing things for, over 20 years or whatever, it's over 30 years. He instead wanted to take a genotype first approach. And so the idea was that he would make a list of genes that he thought might be in some way likely to be mutated in a autoinflammatory disease. And then he would use that list of genes to interrogate a whole bunch of genome sequences or exome sequences of patients. And so rather than just coming up with a random list of genes, he came up with all of the genes that have something to do with ubiquitination, which is a process of post-translational modification of proteins. We had found a couple of genes by a phenotype first approach that involve ubiquitination that defined new autoinflammatory diseases a year or two before he came to the lab. So ubiquitination, okay, that's all right. There's 841 of those genes, so it's a lot of genes. And then he took a set of exomes and genomes from our cohort and from the Undiagnosed Diseases program. It added up to 2,500 exomes or genomes. And so he screened the 841 genes against the 2,500 exomes and genomes, looking for something that could define a new disease. And in doing that, he found 3 patients who were middle-aged to elderly men who had mutations in this gene UBA1. And it seemed that they were heterozygous, that they had one normal copy and one mutant copy of this gene, which was a little curious because you see this gene is encoded on the X chromosome and men only have one X chromosome. So how can these guys be heterozygous? So could be a sequencing error. It wasn't. Could be that they have an extra X chromosome. They didn't. And so then the other possibility is that it could be that it's a somatic mutation. And in fact, that only a subset of their cells in the blood that provided the DNA had the mutation, and that rather than all of the cells having both versions of the gene, that there'd be some of the cells that would have the bad version of the gene and some of the cells that would have the good version of the gene. So he tested for that, and it turns out that's true, that in fact, the lymphocytes have the good version of the gene and the myeloid cells have the bad version of the gene. So it's somatic mutation. So there are 3 things that are surprising about this. What's not surprising is that a young person with a new idea could actually be right and change the way we're thinking. But the 3 things that I think are really important from this are, number 1, that he was able to succeed using this genotype first approach. Number 2, that this is an example of where you actually have somatic mutation causing an autoinflammatory disease. Somatic mutation is a common cause of cancer in adults. So maybe we need to be thinking about somatic mutation more with regard to adult onset autoinflammatory disease, which has always been a very tough nut to crack. And then the third thing is that once we have a broader appreciation of who has UBA1 mutation, it turns out that there are people that carry clinical diagnoses that go across a broad spectrum. So maybe, in fact, our taxonomy of disease is wrong. and that the clinical names that we put on things are not really the best, and that there would be another set of names based on functional genetic abnormalities that would be a more logical way of organizing human diseases. So fantastic. It was a surprise, but I'm delighted to be surprised and delighted to be, in some way, proven wrong. That's it makes the world go round.
Amazing. So Dan, what's the future of autoinflammatory diseases as you see it?
Well, the future of autoinflammatory diseases is the young people who are working in the field. The David Becks, and there are many, many of them out there who have great new ideas, who are excited about doing the same thing that we've been talking about for the last little while, of combining clinical medicine with basic research and trying to use that connection to advance our understanding of human disease and to advance treatment of patients.
And with that, it sounds like you're sort of passing the torch. So what's in store for Dan Kastner in the future?
Well, I will confess that I'm 71 and a half years old at this point, and I am a human being. So, I'm not going to last forever. But for the time being, I am delighted to continue to have my research lab and clinic at the NIH. I stepped down as scientific director of the Genome Institute a little over a year ago, so I have more time for patients and for the lab, and I'm going to keep going as long as I can.
That's amazing, Dan. And it's been so wonderful to have you as one of the people I look up to when I think about medicine and research and clinical care. I think your focus on patients and research is really inspiring. I know that I can say not just for me, but for many of us out there, especially those of us interested in autoinflammatory diseases. It's really been a pleasure to know about you and certainly get to know you in the few interactions that we've had. So I have to thank you from all of us. It's great to have you in our field, Dan.
Well, John, thank you very much for those kind words. And it's really great to have you as one of the people who will definitely write the next chapter in the autoinflammatory book.
Awesome. Thank you, Dan. Have a great day.
You too, John. See ya.
Well, there you have it. I'm ready to follow in Dr. Kastner's footsteps and discover some new diseases. Are you? Thank you all for listening, downloading, subscribing to our show, and for rating us with 5 stars, or however many stars you think we deserve. You can find our show notes for this episode at acronair.org, as well as Apple Podcast, Google Podcast, and on your favorite podcast app. Until next time, please keep advancing the field of rheumatology. And as always, stay healthy.